4 continuation. This session is supposed to chaired by Dr. Thierry HEULIN. He is from France, but he will join us on Zoom. He could not come due to some unavoidable circumstances. So there will be 4 papers. We try to finish this 4 papers on time. That means 15 into 4, 1 hour, and then we have 15 minutes for discussion. So let us start with this Zoom connection with Dr. Thierry HEULIN, CNRS France. Please, Zoom connection.
For the prediction of climate change, this is the IPCC report, the last one. Different scenarios are possible. The more optimistic, I will start with this one, is if we have an extensive use of negative emission technologies, we can expect a decrease of CO2 in the atmosphere. If we have a gradual censoring of current policies, also a decrease, not at the same extent, but with moderate action, we will have an increase of the CO2 emission. And if we have the current policies, this is a very dramatic situation with 3 times the concentration of the emission of CO2 as actually. So, and you can see in this part that the prediction are not exactly the same for nitrous oxide and sulfur dioxide. So the Intergovernmental Panel of Climate Change, the IPCC, the last report, the report of the United Nations leading climate change experts sounds the alarm. Even with global warming limited to 2 degrees by 2050, the consequences will be cataclysmic. The figure they put forward are very worrying. The central figure of this report is global warming compared to the pre-industrial period. The IPCC used the 2 assumptions, plus 2 degrees by 2050 is the ceiling set of the Paris Climate Agreement, but plus 1.5 degrees is the largest set by the countries that signed this Paris Agreement in 1915. The average increase observed is currently 1.1 degrees Celsius. The IPCC also estimates that with a warming of more than 2 degrees Celsius, the melting of the Greenland and West Antarctic ice sheets could be a point of no return. These ice sheets alone contain enough water to cause 13 rise in sea level, meter rise in sea level. In a previous report, the IPCC estimated that sea levels would rise by 1.1 meter by the end of the 21st century. And the IPCC points that 4 to 10% drop in the population of the main crops over the last 10 years. How to capture CO2 to save the climate? Different solutions: reforestation, afforestation, collection and carbon storage by using bioenergy, biochar, carbonates formation, underground injection of CO2, ocean fertilization. But this is no longer possible. And at the end, carbon storage in soil. This is the largest potential of CO2 extraction and lowest cost per ton of CO2. What is the 4 per 1,000 initiative? this is a global network for international cooperation on accelerating the adoption of best practices of agriculture transition. Why storing carbon in soils? To mitigate climate change through the attenuation of GHG emissions. Organic carbon can be stored in soil by decades to millennia. This is also a low-cost negative emission technology. This is an important point to adapt to climate change, because soil organic matter increase water retention capacity by soils and decrease the sensitivity to erosion of the soil. And the end, to contribute to food security and restore degraded soils. Major role of soil organic matter in soil fertility, yields stability, and also restoring degraded soils. So where does it come? Where does it come from? This for 1,000. As you can see, the idea is to store this 4 gigaton of CO2 in the soil. The deeper the storage of this carbon in the soil will be important, the more stabilization of the soil will be efficient. So this is the net emission of CO2 from fossils use, considering the emission and the storage, including ocean, but we cannot increase the ocean storage of carbon. The idea is to maintain the storage of carbon in the ocean. The only way to increase the CO2 is to increase this CO2 in soils. So the goals of this initiative is to increase carbon sequestration in soil as organic matter for increasing good food security, adapting agriculture to climate changes, and to mitigating climate change, pursuing the objective of the sustainable development adopted by the United Nations. This is, you know, that there's a different item of sustainable development goals. The targets of this initiative in relation of these sustainable development goals, you can see here the number 2, zero hunger, the 15, life on land, and the 13, climate action, and the 6, the 12 and 15 are also sustainable goals, development goals targeted by this initiative. About 500 signatories of the Paris Declaration. There is a consortium members, already more than 200 signatories of the Declaration of Intention number 2. The chair is Dr. Ibrahim Mayaki, and the vice chair is a former Minister of Agriculture, Minister of Agriculture in France, Stéphane Le Foll. An overview of the partners and members, you can see that there are a worldwide repartition. And the members are in red, in green, and the partners also in more intense, less intense green here. And you can see that Bangladesh could be one of the future members of this initiative. This is my hope. So you can reach this initiative using this web address, and you can have all the information. So the priority is to increase sustainability carbon storage in cultivated soil and in depth. Why the priority is to increase this carbon storage? Because beyond the urgent need for atmospheric CO2 mitigation, there is also a crucial need for increasing soil carbon content, not only in the first centimeter of the soil, but also in depth, to prevent soil degradation and to increase soil fertility. Why choosing photosynthetic active plants, crops, rather than plant residues? Because we use the fact that plants are transforming CO2 from the atmosphere in photoassimilates, and these ones are allocated to different parts of the plant, and especially as the root exudates, very rich in carbon, but this carbon is unstable. And to transform this unstable active carbon, some bacteria producing isopolysaccharide are able to transform this unstable active carbon, such as glucose, for example, or fructose, in bacterial polysaccharides that are more stable carbon. Especially when these polysaccharides are associated to minerals. And at the end, we have a soil aggregation due to this bacteria, and the soil aggregates are also stick to the root system. We have done some experiments using these phenotypes, the root adhering mass divided by root biomass as a phenotype of root exudation. There is a very good correlation between the quantity and quality of root exudate and the production of this root adhering mass. We have done a GWAS analysis, and we have some potential gene candidates to explain that this phenotype is also controlled by the genetic, under the genetic control. So we have to test the candidate genes. So root growth and root exudation are potential adaptive traits for abiotic stress tolerance in pear millet, and for root prospection in depth and carbon storage in soil with very low carbon content. Is plant breeding a possible way to increase carbon storage in soil? Yes. If phenotyping of root system and rhizosphere is possible, especially if this phenotyping is possible in natural conditions. So, to our opinion, there is an urgent need for in situ root phenotyping. As a conclusion, we are on the way to get a proof of concept that plant breeding based on root exudation and root development in depth is a promising way to increase soil organic carbon. This increase of soil organic carbon is crucial for preventing soil degradation, increasing soil fertility, and plant productivity. Even if the effect on atmospheric CO2 mitigation is limited, and not the first objective of farmers, underground-based plant breeding will have a direct and positive effect of human food supply. So, as a summary, it is very clear that climate change have an effect on agriculture, and the question is agriculture changes are able to have an effect on climate change through the mitigation of CO2. Thank you for your attention, and if you have questions, I'm ready to answer to it. Okay.
Thank you very much, Thierry. We don't know, audience can say whether we changed agriculture or changed the climate. This is the question. So as Thierry HEULIN is online, so if you have question, we can finish it here.
Yeah.
Yes, please. Tafadjal Hussain from Bangabandhu Sheikh Mujibur Rahman University.
Please ask the question very slowly because the connection is not very good. So please.
Thank you so much for an exciting presentation and thought-provoking presentation. You mentioned that we should focus plant roots phenotype for breeding strategies. In that case, you see our major crops, rice, wheat, maize, they are not deeply rooted crop. So in that case, and we must need to ensure the food security considering the climate change and other issues. So food security is the prime issue. As you mentioned, population is increasing and it would be, 10 billion by 2050. So in case of major food crops like rice, wheat, maize, what kind of food traits we can consider along with, grain yield and others for considering the higher carbon sequestration and lower emission of carbon to the atmosphere?
I think there is a compromise to be searched between the production of grain. Of course, you have to crop rice, maize, wheat for sure. But our feeling is that it is possible to have a very good compromise between the plant breeding focus on the grain yield and the input, the root exudation and the possibility of for the soil microbiota, the microorganisms in the soil, to transform the root exudate in a more aggregated soil. So I have the feeling that I know the field in Bangladesh. The question of the water supply is not the major question, but the stabilization of some soil in Bangladesh will require an increase of soil aggregation in some situations. So at the end, we have, for the crop breeding, we have to find a good compromise between selection, plant breeding focus on the grain yield and also on the aggregation of soil in the rhizosphere.
I have
yes,
a question.
please, Professor Karim.
Thank you very much for a very nice presentation. I have one question. Bangladesh is a highly densely populated country. And our forest land is around 15%. That should be about 25% or more. But our farmers, for their survivability, they have to clear sometimes the forest. And by clearing, they also grow the crops. For example, they clear the forest, but around the year, they grow different crops. Do you think by clearing the forest and by growing the crops, for example, rice, wheat, or even maize, C4 and C3 plants both, do you think that will compensate to some extent the carbon sequestration or other matters of the
Yes, again, dealing with the forest is quite clear that most of the forests are on soils very rich in carbon. My talk is especially, yes, the strategy to increase the soil carbon in soil are mainly in relation with soil with poor carbon contents. The problem is that under forest, the soil is more or less, in Europe this is the case, the soil under forests are very rich in carbon. So when you transform the forest in crop, you have a very good situation for the production, but at the end, for the carbon storage, it's not a very good idea. So my opinion is to try to not to limit the deforestation for sure at the scale of the world. This is the main purpose. I mentioned this point. We have to stop the deforestation. But I understand that in some countries, such as in Bangladesh, sometimes you need to increase the crop superficie. So again, we have to find a good compromise between the change of land use and the possibility by this way to increase the carbon storage in soil. But your question is very difficult to answer.
Thank you, Thierry. See you soon.
Thank you. Yes, see you soon. Thank you very much.
Thank you very much. So now I request Professor Muhammad Manjurul Karim to present his paper. Entitled his paper, Potentiality of Using Salt-Tolerant Plant Growth Promoting Rhizobacteria as a Biofertilizer for Climate-Smart Agriculture in the Coastal Areas.
Okay, am I audible? Yes. Okay, Assalamu alaikum, and I would like to present my talk. Before that, I would like to thank the organizing committee for this international conference on climate change and food security. My humble thanks to the chief coordinator, Professor Dr. Mustafizur Rahman, and his team. So as you can understand and rightly pointed out by the chair of this session, the title of my talk is Potentiality of Using Salt-Tolerant Plant Growth Promoting Rhizobacteria as Biofertilizer for Climate-Smart Agriculture in the Coastal Areas. So as you have pointed that some of the keywords being highlighted, salt-tolerant, PGPR, and climate-smart agriculture for coastal areas. Right. First of all, here is the data presented, offered by the International Geosphere Biosphere Program, where it is shown that we are having an increasing atmospheric carbon dioxide and also an increment experienced in the form of global average land surface temperature. Of course, we are having melting of Arctic summer ice and eventually the global sea level, rising of the global sea level. All this data cumulatively piling up the climate change index, making the climate change a fact, not a fiction. One major challenge for the 21st century will be the production of sufficient food for the increasing global human population. This has become a threat to the existence of our planet and can potentially harm the food security in the form of land erosion and salinity intrusion. So here we are presented being the world's largest deltaic basin, and this is the part of a glimpse of Patuakhali, the district in a village called Pakhimara in the dry season. This is really a very salt headed cultivable zone with no cultivation. So this is a real problem, a very climate change induced problem. What is the way out for a sustainable development? Well, bioinnovation and bioeconomy is being pointed out, whereby we use development of salt-resistant crop varieties. So this is really been gaining a momentum, but its absolute acceptability is being questioned due to its ethical and environmental issues. So the plan B, as I'm pointing out here in this conference is microbial technology by introducing suitable application of salt-tolerant plant growth promoting rhizobacteria. Why? Because it supports profusely the plants directly as well as indirectly to withstand the stress. Maybe it could be salinity, it could be drought, you name it, we have got the natural solution. So, this is a completely green approach to ensure a sustainable agriculture. So, this is how I have, structured my talk, starting from the sample collection, definitely from an area where plants are being grown under a salt stress condition, definitely in the coastal areas. As we'll be sampling rhizospheric soil, we also attempted to analyze the geophysical parameters of the sampling sites. And the bacteria from the plant rhizospheres was isolated. It was first tested for the halo tolerance, and then the several in vitro assessment of plant growth promoting traits. We try to address the biofilm detection through scanning electron micrograph to ensure that the conglomeration of the bacteria by dint of production of biofilm ensures their better survival. Then we did some in-plant experiments, and finally we tried to address the molecular crosstalks for stress management. So here we go, this is the sampling sites. The sampling sites definitely encompasses two different zones. One is the saline zones in the district of Patuakhali. We held sampling in 4 different upajilas of the Patuakhali districts, and we also sampled from a non-saline zone, which is Choddogram Kumilla. As you can understand that the EC of, the electric conductivity of the saline soils appeared greater when you compare with the non-saline soils. It was close to 7.8, the electric conductivity of the saline soils, as opposed to only 1.7 for the non-saline soils. And this is well correlated with the higher pH. As you can see, this is the higher pH. You can't see the Y scale, but this is the higher pH, as you can see. And this higher electric conductivity ensures greater level of sodium ion as well as chloride ion content, as opposed to the non-saline soils, where there is a marked reduction of the sodium ion as well as the chloride ions. These sampling sites I have chosen for collecting the samples from the root rhizospheres, and this is the site where already rice is being cultivated. So I prefer to sample the root rhizospheric soils for the collection and isolation of salt-tolerant bacteria. So these bacteria, which I have been collected, first tested for their salt tolerance, and on an in vitro system in a plate composed of different level of sodium chloride level, and you understand that I was able to collect a good number of salt tolerant bacteria having 1.7 molar salinity stress capability, which is close to 10% of salt, and this enables us to further characterize those bacteria. One important thing is that I made this plate selective by not adding any nitrogen source. Thereby, the plants, thereby the bacteria which is appearing on this media are definitely nitrogen fixers. So this is one, after understanding that these bacteria can fix nitrogen, their ability of nitrogen fixation was estimated by Zeldal method. We also tested with the phosphate solubilization method, phosphate solubilization ability, and indole acetic acid a phytohormone, an auxin, which is important for the plant growth. So these bacteria, all these PGP activities were analyzed, and we tried to figure out, we tried to shortlist which are those bacteria that can cumulatively, okay, cumulatively confer all these properties within themselves. So here is the, a 3-in-1 figure where I showed indole acetic acid production by the bluebirds, phosphate solubilization by the redbirds, and the green triangle shows the nitrogen fixation. And after analyzing more than 59 isolates, we able to shortlist at least 4 bacteria, which was eventually sequenced, identified by gene blast from the NCBI database. And these bacteria were Bacillus aryabhati, Acrobacterium intermedium, and Achromobacter denitrificans. Out of these 4, the next experiments will engage Bacillus aryabhati for the in-plant experiments. Before I go for the in-plant experiments, I had a chance to look into this bacteria, what makes them aggregated under stress. So as you can see on your right-hand side, this is the bacteria found from the non-saline isolates or non-saline areas, and the bacteria which were isolated from the saline zones. We found that there is a clear EPS layer outside of the cell surface, making them aggregated in the form of biofilm and withstand the stress. So in the in-plant experiments, I used a salt-sensitive rice cultivar. I repeat, this is a rice cultivar BD28 of Oryza sativa, and this is a salt-sensitive. We did a good number of seed germination and biofertilizer preparation. As you can see, the seeds of a salt-sensitive cultivar germinated by thermal method, which were later floated on hydroponic solution. After 15 days of cultivation in hydroponic solutions, plants were transferred to earthen pots, and biofertilizer F, as you can see in the figure F, was prepared with isolates of interest, hereafter Bacillus aryabhati. So we found 93% of seeds were germinated, of which 89% survived in hydroponic solution. So these were eventually carried out on earthen pots. So I would like to, before I describe these results, I would like to catch your attention by saying that 2 far left areas, 2 far left pots, these are control pots with no inoculum, and 2 right, farmost right pots, these are plant pots which was supplemented with E. coli DH5 alpha, and this is the bacteria known to have no plant growth promoting activities. So this was acting like a control, negative control. And the pot number 3 and 4, this is the pots of interest where I supported with the biofertilizer. So till 45 days of biofertilizer application, we found that 80% of the plants able to grow without support of biofertilizer. If supported with the biofertilizer, MS3 hereafter, it was 90%. Now, I exposed those pots with the salinity stress after 45 days with 200 millimolar salt. And after this stress, the picture changes radically. As you can see, only less than 5% of the plants able to withstand the stress without the support of biofertilizer, and the plants which were supported by biofertilizer able to withstand the stress up to 45%. So this makes the preliminary observation that the MS3 hereafter the biofertilizer somehow helping the plants to withstand the salt stress. So what is the phenotypic changes that we have noticed? And the changes, as we can see, the blue birds are the no inoculum, red birds with the E. coli, and the green birds with the MS3 biofertilizing inoculant. And in all those experiments where I tried to analyze the different phenotypic traits starting from the dry weight, carbohydrate content, you see that the taller green bars in each graph denote better support to rice plants provided by biofertilizer MS3 under saline conditions. Next, we addressed another traits, and this is called proline. You know the proline is a compatible solute. Accumulation of proline gives the cells an edge to withstand the osmotic stress. And you see that the plants supported with the MS3 biofertilizer are having greater amount of proline content, okay, the green bars hereafter. Another thing is the MDA, malondialdehyde. This is another metabolite. The cells that accumulates high MDA indicates membrane lipid peroxidation, sustainability, then sustainability of plant growth under high salinity is associated with reduced MDA formation, which means once you have reduced MDA formation, you have better chance of survivability, and you have, and the vice versa. And definitely the plants supported with the MS3 have reduced in the green bars, and then in the green line you see it has the reduced MDA and supporting the plants for withstanding the stress. Next, we attempted the gene expression, what makes the plants survive? What is being going on inside? What is the crosstalk between the plant and microbe? For which we attempted to study at least 4 different salt tolerance genes from plants, starting from the NHX1, GIG, BZ8, and SOS1. These are the salt tolerant genes believed to confer salt resistance to plants. And we are trying to see the level of these genes after addition of biofertilizer, the MS3. For this, we attempted to do a semi-conservative reverse transcriptase PCR, where we collected the tissue samples from the plants. These were, the total mRNA was collected. By a RT-PCR, this mRNA were converted to the cDNA, and in this cDNA, we did a PCR for those salt-tolerant genes and did the agarose gel electrophoresis. This is very important. As you can see, you need to understand that the none versus MS3. This is the black bars, whereby you see that upon addition of MS3, the expression level definitely goes up, as you can see in the closed bars. So this is under regular condition. When you change the condition from the non-salinity to the salinity of 100 millimolar, you see these are the 2 green parts you need to compare, and we found that the level of expression in plants supported by MS3 went up when compared to plants not supported by MS3. So this is the thing, that the salt resistant genes in plants somehow got overexpressed in the presence of bacteria supporting the plants. As an internal control, we used eukaryotic initiation factor 2 alpha. No matter whatever the conditions, their level of expression didn't change. So can I have 1 minute? Okay, so this is a cartoon where I tried to figure out that plants suffer salt stress and succumb to die, but if they are populated with the PGPR, the stress are being channeled into the vacuoles and giving an edge the plants to survive under the stress. In conclusion, we can say that efficient strains could support arrays of PGP activities under salt stress. therefore are potential candidates for biofertilizer preparation. And resistance to salts, antibiotics, and heavy metals. I didn't show the data for antibiotics and heavy metals, but I was able to figure out that they appear to contribute by the biofilm formation of an EPS layer outside of the cell surface. And finally, the stimulation by PGPR as a mechanism of inducing salt tolerance in rice by modulating differential transcription of a set of salt-tolerant genes. So this is my research experience whereby we try to figure out to make the vicious cycle can be well coordinated and regulated if we able to implement the PGPR, and definitely it would give us the golden harvest. So take home message is the suitable application of salt-tolerant PGPR can be harnessed to bring the coastal agriculture into sustainable crop production. So these were some of my glimpses of the papers being published in Canadian journals as well as other frontiers and papers. And finally, I would like to thank my collaborators, Dr. Tariq Rahman, Dr. Sirajul Haq, Dr. Zeba Siraj, and my students and Ministry of Education for funding. Thank you very much for your patience hearing.
Thank you very much, Muhammad Manjurul Karim. We will wait for discussion after finishing the presenting paper. So now I would like to invite Abdul Karim to present his paper.
Anyway, my talk is on challenge of climate change and strategic research on Bangladesh agriculture. You know, in agriculture, Bangladesh has made a big leap, big success, and especially in cereals, we are self-sufficient, we claim. Though for other crops, the scenario is not so promising. But the problem is, every year, our population growth is 1.7 million. That means 17 lakh. And for them, we need extra 300,000 ton per year food. On the other hand, the land is decreasing at the rate of almost 0.7%. So these are the big challenges. How far or how long we can support our population, we can feed our population. So according to IPCC 2022, that is AR6, 14 hazards have been identified. That is monsoon rain, flash flood, drought, salinity, storm surge, excess heat, landslide, river erosion, lightning, excess rainfall, cold spell, sea level rise, urban flood, ocean acidification. So these are the IPCC prediction. And what will be the consequence in Bangladesh? That is, the temperature may rise. The temperature will rise. In that case, the sea level will increase. And it is predicted that even 1 meter increase in sea level may inundate 18% of the coastal region, and may 30 million people may become climate refugee. And salinity, of course, several speakers talk on salinity rise increasing in Bangladesh, and also our drought in the north eastern part, that is marine tract especially. It is drought affected and rainfall is decreasing there, temperature is increasing, and flood or water logging, that is already 2.6 million hectares, water logging 0.5 million hectare, and chore land. We have already 1 million hectare chore land. So these are the concern, these are the impacts of climate change, and which will be increased further. And sea level rise, so many prediction that even 1 degree, as I already told, 1 degree rise in temperature may inundate 18% of the people. But according to Bremer, I think soil scientists, they know Bremer. He worked in Bangladesh for maybe more than 20 years. But he has published one paper in 2014. He showed that the land in Bangladesh is increasing, especially in the south. And last month I went to in the south coastal area, Subarno Chor and Hatia then, and I heard there is a new chor is developing there, though still it is not cultivable, which is 100-meter long. So these are the, according to Bremer, in Bangladesh every year almost 19 kilometer square land is increasing. So these are the, sometimes we say, though climate change, it affects many parameters or it is harmful, but on the other hand, due to climate change, our land is increasing because the Himalaya soil, in summer, I'll talk later on also, in summer we get a lot of siltation that increasing our land, also productivity of our chor land. So these are the benefit of the, climate change issue. Though there are a lot of harms, but there are benefit also. I want to make a joke that Gopal Bhar. There, I should talk in Bangla for the joke. Gopal Bhar, একটা আলুর দোকানে ওই আগুন লাগছিল, আগুন লেগে তো মহাজনের তো মাথায় হাত, কিন্তু গোপাল ভাঁড় গিয়ে সেখানে আলু খাচ্ছে। তারপর মহাজনকে জিজ্ঞাসা করতেছে আবার কবে আগুন লাগবে। সো বাংলাদেশের যে এই যে ক্লাইমেট চেঞ্জের ইফেক্ট, এই যে বড় বড় ফ্লাড হচ্ছে, এর কিন্তু বেনিফিট কিন্তু আমরা পাচ্ছি, যত বড় ফ্লাড তত কিন্তু বেনিফিট পাচ্ছি আমরা। তার মানে সেই গোপাল ভাঁড়ের আলু খাওয়ার মতোই যে বড় ফ্লাড কবে হবে, কারণ এটা আমাদেরকে অনেক advantage পাচ্ছে না। Salinity already আলাপ হয়েছে, that already salinity is increasing in the south and almost 29,000 square kilometers affected by salinity. But the I would like to talk some positive side also. The salinity increases and the very high extent of salinity usually in March, April, when is March to May, when is Kharif one season, and we don't have so much crop in Kharif one season. But in summer, the salinity goes down. So we can grow almond rice easily in the south, even in the coastal region. And in the winter, as the winter season starts from November and continues up to March, so our winter crops doesn't face too much the salinity. So our challenge to establish crop in Kharif one, that is our challenge, of course. But other seasons is not that much problem, I think. And what are the causes of this salinity? First problem, I should say, geophysical location. Because we are the foothill of the Himalaya, and our river originates from other country or from Himalaya. So the water is diverting now. So we have a lot of siltation. Every year we have 2.5 billion tons sedimentation comes from Himalaya, and that making a barrier on the river, and that makes barrier on the flow of the water also. And of course, some anthropogenic activities, for example, shrimp culture, or some making the polder, this kind of things, also increasing the salinity. But some the technologies are also available. For example, the relay cropping or shotgun technology, or growing lentil or grass pea within the strip of the almond rice at the later stage of before harvesting the almond rice. So these are the technology already we have, and also these are the shotgun. Already I think someone talked that we can grow crops on the strip as well as we can grow fish or the water-loving plants, for example. We can floating agriculture in Bangladesh. It is very popular. So we are using the land for this purpose. And then also some salt-tolerant mung bean, soya bean, or rice already we have. So we are combating the saline problem area. And then flooding is a problem. Flood is I already mentioned that flood is a problem as well as blessing for us. If you see the statistics, after every big flood, our crop production, especially our winter crop production, borrow rice or wheat or other winter crops, pulses, oilseeds, increases. So every big flood, every big floods, we get higher production. So these are the benefit of the flooding. But of course, we have suffering, but that not so very long time. But problem is the flash flood. You grow crops, suddenly the flood come, even it continues for 2 weeks or 3 weeks, but it damages your crops. Flash flood is dangerous than our regular flood. Regular flood is not a big problem, I think. So flood is very common where two rivers meets. For example, in the Kurigram or in the Chandpur area, or in the where the Poddayan in Maniganj, near Maniganj. These are the area where flood is a problem. And then I already mentioned that we have almost 1 million hectare chol land. And this productivity of chol land is increasing day by day because of the siltation. And also the productivity of chor increases with the age of the chor. The older chor has higher productivity. And fortunately, our chor land, or unfortunately, of course, the river, it makes barrier on the river flow. But fortunately, our chor land is increasing. And farmers, Bangladesh farmers are so smart that they grow 3 crops or 4 crops at the same time. My cropping system is very high. Cropping intensity is very high. They grow 3 crops. Even in the Tista belt, I went to Tista, even on the Tista river, they can grow 4 crops at the same time. So they are not idle, they are using it. The concern is that suddenly the water comes from the other side. Suddenly that damages the crops. Otherwise, if the farmers say, if the water doesn't come, then we can grow the crop around the year in the chol land. So chol land is I think we get advantage. And then of course we can grow our in Bangladesh, you know, now when the India or even Bangladesh stops importing Indian cow, then our cow production, our dairy production increases. So I think Bangladesh government should stop. There should be no cow from India. I think then our farmers will make self-sufficient in dairy or cow production in Bangladesh. Some problem, you know, landslide is a problem in the chor land. Even we know 100 years old chor land may anytime may disappear. And that flash flood damage sometimes the crops, it is true. But we have technology for deep water rice. I don't go in details. But we have one technology. We said if the flood especially in the northern chor, if the flood continues for more than 3 or 4 weeks, there should be some seeds we call insured seed bed. That means there should be some seeds, and that is the government job of the responsibility of the government. They should supply the seeds so that farmers, they can grow seed bed, floating seed bed, so that they can go for second time planting. And some research are going. This is the picture of Bangabandhu Agricultural University. Already we have identified some flood tolerant material side by side with that Viridian 52. We have also identified some materials. Then we are also some mung bean, that is waterlogging tolerant mung bean, we have identified. Then drought, that is the northwestern part of Bangladesh, this drought. The area is, you know, almost 77,207 hectares. It's a big land, where the problem is the temperature is much higher than the national average, and rainfall is much lower. In one side, temperature is much higher. On the other side, rainfall is much lower. That is the problem in the rain-fed area. Our research are going on screening wheat or some soybean, and we have already identified some materials that are tolerant to drought, and also some technology from BRI. These are the BRI that growing 3 crops: rice, then short-term mung bean, and then again rice. These are the technology in the barren tract. And also the relay cropping, rice and then chickpea or lentil or mung bean even. And this is very interesting technology that a pond of 12 meter into 12 meter and 3 meter deep can save more than 1 hectare of crops. This pond, they catch the rainwater and then keep, and when the crops suffer, especially at the latter stages of its growth, when the suffers from the drought, then this pond can use it for supplemental irrigation. So that can save at least, one pond can save at least one hectare land. So this is very useful, and this is going on, this practice. Why agricultural production is increasing? Because of this kind of small technology is practicing throughout the country. Even in the Satkhira, saline zone, they catch the rainwater by making this kind of pond, and then they use the rainwater for the crops. This way they reduce the salinity. So these are technologies, these are the reality, these are practicing in Bangladesh. And seed priming is a technology, just you soak the seeds with water for 6 hours, and then you sow the seeds, then the germination enhances. And once the seedling germinates, the roots goes to the deeper zone, then it doesn't suffer that much from the drought. Mulching, of course, conserve the soil. And few soybeans, I don't like to talk much about soybean now. And then our research, what should be our research? The appropriate cost-effective technology. That is of course, yesterday I had a discussion meeting at BRC. Our research thrust is based on the hottest spot. That is drought, that is salinity, that is Howard, that is hill, this kind of chore land. So the technology should be target oriented, because we have to address this hot spot that is suffered from the climate change. And lab to farmers concept, many times I talked with Professor Tufajjal, because he's biotechnologist. We say it's biotechnology should the development should come to the fore for our eat, from DNA to our food, for safe food. So lab, field, and then table. This should be the molecular biologists, physiologists, biotechnologists, and then agronomists, soil scientists, even nutritionists. They have to, the work should be concentrated, comprehensive, collaborative work, that should be. And then conclusion. I come to the point because time is very short. The chairman is looking at me. I don't know why many times he look at me. So anyway, so the challenge is that our population is increasing, as I already told you, 1.7 million per year. Our land is decreasing almost 0.7 million, so 0.7 million ton hectares. And then climate change, this challenge. So these are the challenges. And what to do? The technology we have developed in the university or at the research table, but finally that technology should be fine-tuned, or before releasing technology, farmers should be involved. That means that should be participatory approach. We have developed more than 100 rice. Why in the field only we get 10, 20 varieties? Why? Because the farmers were not properly addressed before developing the variety. So whatever the technologies, maybe microbiology or whatever the technology before development or before application, farmers should be involved. The research should be participatory, involving the end user, the farmers. Thank you very much, Mr. Chairman.
Thank you very much. We are going to the next paper very quickly. Marufa Afroz from Bangladesh Agriculture Research Institute, Gazipur, Bangladesh. So this is the last paper of today's session.
Honorable Chairperson, respected foreign delegates, dear audience, my dear colleagues, Assalamu alaikum and very good afternoon, everybody. I am Dr. Mafrur Afroz. Welcome all of you to my paper presentation session. The title of my paper is Differential Colonization of Solanum sicimbrifolium and Tomato by 8 strains of Ralstonia solanacearum. Bacterial wilt, which is caused by Ralstonia solanacearum. Actually, host strains, their host strains change within that time, and finally, Uyghur et al. Found that more than 450 plant species belonging to 54 families, and it is found tropical, subtropical, and warm temperate regions. So, similar like symptom, this is brown rot of potato, in tobacco, granule wilt, Moco disease in banana. Now, Ralstonia solanacearum, I would like to say something about this because initially Pseudomonas solanacearum, beta proteobacteria of Burkholderia group, gram-negative rod with a polar flagella, oxidase positive, arginine dehydrolase negative, accumulates polybeta hydroxybutyrate. Ralstonia solanacearum species complex. So there are many bacterial wilts and there are many Pseudomonas solanacearum. Ralstonium CG have high DNA homology, non-flagellate, blood disease bacterium, initially Pseudomonas syllabensis, now called BDB. Genetically close to Ralstonia solanacearum, differ in morphological and biochemical properties. Both Ralstonia solanacearum CG and BDB produce specific band with Ralstonia solanacearum specific primers with 759, which is forward, and 760, that is reverse primers. So, Ralstonia solanacearum have 5 races based on their host strains and 5 biovars based on their biochemical properties. So, from this table we found that they have 5 races. Race 1 have wide host strains and their geographical distribution is Asia, Australia and America, and their biovars 1, 3 and 4. Race 2 contain bananas, other musa species, and their geographical distribution Caribbean, Brazil, Philippines, and their biovars 1. Race 3 contains potato, geranium, some other solanaceae, plus a few other species, worldwide except USA and Canada, geographical distribution, and their biovars 2. And race 4 contains ginger, host strains, geographical distribution Asia, and biovars 3 and 4. Mulberry, race 5, and their geographical distribution China, and biovars 5. So based on utilization of a set of carbohydrates and alcohols, they have 5 biovars. Those are the carbohydrates and those are the alcohols. So biovar 1, biovar 2, biovar 3, biovar 4 and biovar 5. So they have 4 phylotypes: phylotype 1 Asia, phylotype 2 America, phylotype 3 Africa and island in Indian Ocean, phylotype 4 Indonesia, Australia and Japan. Sequivers, endonuclease, gene sequence variants 52 and increasing. Clones, DNA fingerprinting techniques such as REP-PCR, box, REP, ERIC, PFGE, several. So actually I would like to say in a summary that species Ralstonia solanacearum species complex, which is a specific primer using the primer 759 and 760 phylotype. There are 4 phylotype using ITS multiplex PCR, sequivers phylogeny with or specific PCR, and clonal lines PFGE, REP-PCR and AFLP. So major problem for vegetables and crops such as tomato, eggplant and pepper in South Asia. And Solanum sicimbrifolium, sticky nightshade, fire and ice plant, lychee, tomato, etc. So previously I said that Ralstonia solanacearum causes vascular wilt in over 450 plant species belonging to 54 families, especially solanaceous crops. It is an economically significant pathogen of tomato, potato, tobacco, eggplant, banana, groundnut, ginger in tropical environment. Bacterial wilt is a major constraint for production of eggplant, tomato and pepper in South Asia including Bangladesh. Host resistance is the most practical and sustainable approach to manage the disease if resistance variety are available. Grafting of commercial varieties of Solanaceous vegetable crops onto resistant rootstocks is an acceptable approach to combat bacterial wilt caused by Rhizosphere solanacearum. Bacterial wilt resistant wild solanum is a popular rootstock in South Asia. Plants grafted onto Solanum sicimifolium not only reduce the incidence of bacterial wilt but also increase marketable yield even in the absence of disease pressure. Under the above circumstances, the investigation was conducted with the following objectives: to determine differential colonization of Solanum sicimifolium by 8 strains of Rhizosphere solanacearum from Bangladesh and Nepal, to compare relative colonization of a wild Solanum species, Solanum sicimifolium, and tomato by a strain of Rhizosphere solanacearum. So those are the materials and method. I will not go to all of these. Experiments, designs and analysis of data. Experiments are conducted following completely randomized design with 4 applications. Collected data are analyzed statistically with SAS version 9.1 using a mixed model and Minitab. Differential colonization of Solanum sicimifolium by strains of Rhizosphere solanacearum. Objective to determine differential colonization of Solanum sicimifolium by 8 strains of Rhizosphere solanacearum from Bangladesh and Nepal. So the first experiment was conducted to determine differential colonization by 8 strains of Rhizosphere solanacearum, and description of the collected strains of Rhizosphere solanacearum are shown in table 1. Those 8 strains, these 7 strains which are collected from Bangladesh and these 2 which are collected from Nepal. So inoculum suspension of Rhizosphere solanacearum are prepared from 48-hour cultures on casaminopeptine glucose agar medium incubated in an incubator at -20 degrees Celsius. Those are materials and method. I will not go to all of those. Actually, bacterial population in Solanum sicimifolium plants are enumerated at 5, 12, and 20 DI. Primers used in PCR for amplifying and sequencing are shown in table 2. Those 2 primers, that is 759 and 760, forward and reverse primer are used in this experiment. Now, colony characters of Rhizosphere solanacearum grown in SMSA media are shown in figure 1. The Rhizosphere solanacearum colony morphology on SMSA media was round, fluidal. The center was dark violet and around the center was pinkish. General PCR with Rhizosphere solanacearum are shown in figure 3. So, lane 1, 3, 5, 6, 8, 9, 10 and 12 gave the positive band and 300 base pair with the positive control of bacterial wilt, that means Rhizosphere solanacearum. Population of Rhizosphere solanacearum is stem base of Solanum sicimifolium plants enumerated by dilution plating at 5, 12 and 20 days after inoculation are shown in table 3. In case of 5 and 12 DI, there is no significant difference among these strains in terms of bacterial population, but at 20 DI, the significantly highest bacterial population was recorded from SM716 and the lowest bacterial population was recorded from SM728. More or less similar bacterial populations are found in Solanum sicimifolium when bacterial populations are enumerated following RT-PCR method. So colonization of Solanum sicimifolium by South Asian strain by dilution plating and RT-PCR method are shown in figure 3. Results presented in this figure showed that bacterial population increased with the age of the plant in both methods at least up to 20 DI. Wilt symptoms 56 days after inoculation with Solanum sicimifolium in order of aggressiveness of different strains of Rhizosphere solanacearum. The most aggressive strain was SM716, which is followed by SM743 and SM730. Area under colonization curve calculated based on log transform value of population of 8 strains of Rhizosphere solanacearum in Solanum sicimifolium by dilution plating and quantitative RT-PCR. In case of dilution plating method, the highest AUCC was recorded from SM716, which is statistically similar with all the strains except SM732, SM728 and SM733. And in case of RT-PCR, the highest AUCC was recorded from SM716, which is statistically similar with all the strains except SM728 and SM733. The lowest bacterial AUCC was recorded from SM 733. Relationship between bacterial population AUCC from Solanum sessimifolium and tomato. The relationship shown in figure 4, the relationship between bacterial population determined by two methods are linear, positive and significant. So, differential colonization of a wild Solanum species and tomato, objective to determine differential colonization of Solanum sessimifolium and tomato by this strain SM 728 of Rhizosphere solanacearum. So, those are the metals method. I will not go all of those things. So, wilt symptoms in tomato at different days after inoculation with Rhizosphere solanacearum strain SM 728. Uninoculated tomato plants are free from visible wilt symptom. Visible bacterial wilt symptoms are observed within 3 to 5 days. Those are 7 DI wilting at 14 DI, and this is wilting at 21 DI. And this is also wilt symptoms did not appear in Solanum sessimifolium within 21 days after inoculation. And those are the pictorial view. So, population of Rhizosphere solanacearum is Solanum sessimifolium and tomato enumerated by dilution plate method and RT-PCR. In case of dilution plate method, the bacterial population ranged 5 to 6.99. The lowest bacterial population was recorded at 14 DI, and the highest bacterial population was recorded at 42 DI. The bacterial population was positively correlated with the DAI. In case of tomato, the highest bacterial population was recorded at 21 DI, and the lowest bacterial population was recorded at 14 DI. The reduction of bacterial wilt may be after 14 DI may be due to severe attack of bacterial wilt and severity of wilt as well as foliage of damage. The strain of 728 was more or less similar in case of tomato and in case of Solanum sessimifolium, both the method, dilution plating and RT-PCR method. So, comparison of dilution plating and RT-PCR to calculate AUCC in Solanum sessimifolium and tomato enumerated as shown in table 6. The AUCC are statistically similar in two crops enumerated by two methods. So, relationship between bacterial population AUCC from Solanum sessimifolium and tomato as shown in figure 6. The relationship between bacterial population determined by two methods was linear, positive and significant. So, relationship between two methods, dilution plate and RT-PCR is highly significant in both crops on Solanum sessimifolium and tomato. There is a strong positive relationship between two methods. Based on findings of the present study, the following conclusion may be drawn. All 8 strains of Rhizosphere solanacearum used in the present study are able to colonize rootstocks, that is Solanum sessimifolium, stem without causing visible wilt symptoms. Quantitative real-time PCR, that is RT-PCR, can be used as alternative of dilution plating method to quantify population of Rhizosphere solanacearum in colonized host tissues. I would like to acknowledge USAID, IPM Innovation Lab, Ohio State University and Bangladesh Krishi Gobeshona Institute, that Bangladesh Agricultural Research Institute. This is all about my presentation. Thanks to all. Thanks for patience hearing.
Thank you, Marufa Afroz, for lovely presentation, and at least there are so many people to ask you so many questions. I don't know whether we'll go for question. You can discuss with her later on, but I can announce you one thing, that you have at least one development of your research that my neighbor Nepal is interested with your work. He's just talking with me. You can talk to him later on, so you can collaborate with him, and he will also like to collaborate with you. Floor is open for any discussion for previous paper also, please.
Thank you for offering me time to ask a question. I truly enjoyed Dr. Manjurul Karim's presentation on salinity tolerant plant growth promoting bacteria, and from isolation to the gene expression of the host. Really fascinating work. And we are also working on plant probiotics in different aspects. My question is, you conducted experiment in the pot. Was the soil sterilized, or you took the soil from the natural, crop field? I didn't get the, soil data because you applied MS3, one Bacillus species. What we feel, as Dr. Karim mentioned, in many cases, up to the pot experiment, we found fantastic results in case of plant growth promoting bacteria. But when we moved to the real field in the farmers, we found very few percentage they persist over there. So do you have any plan to check them in the real field in the coastal area, because your isolates seem very promising? Thank you.
Thank you, Dr. Islam. This is a very fascinating question as well, and a challenging one, from bench life to the real life. Okay, so first of all, the soil that we used from natural soil, for sure, and what we autoclaved afterwards before we conducted the soil pot experiments. So by autoclaving the soil, we made sure that it is sterile. Okay, because I'm trying to point out the specific benefit brought forward by the inoculant to the plants. So that is why it was a customized system through which we can study a specific microorganism of interest. This is a pilot, and definitely in a net house experiment. But we know very well when we go to the real field, surely it will, lots of factors and challenges will plague our findings we found during the pot experiments. So we are recently making a collaboration with a partner from Sher-e-Bangla Agricultural University, and the experiment is underway in the real field. Thank you.
I have one suggestion, I think.
Oh, sure.
Because I also worked with salinity for a long time. I think the word salt-resistant crop, I think this word we should be very cautiously use. Rather, better we use salt-tolerant crops, because the glycophytes, that means the cultivated crops, we cannot find, or we cannot get resistant one. Tolerant, okay.
Yeah, you
So
you nailed it correctly.
We have to be very careful in using resistant for glycophytes or cultivated crops.
Definitely.
Thank you very much.
Definitely. Thanks for your suggestion.
6 o'clock in the evening. We start at 9 o'clock in the morning. So we want to conclude today, but I want to request you to submit your paper, both soft and hard copy, because we will publish it very soon. Okay. And you also request your other colleague who presented the papers earlier in the morning to do this thing. This is one important issue, and I want to mention that this is the second time this conference held in Bangladesh. First time in 2008. After 2008, from this conference, not less than 10 collaborative programs initiated in different places. The conference is, we call, is marketing for the student and marketing for the development collaborative things. So we look forward for those things. So try to understand the philosophy of this thing. So you try to come earlier than today. Thank you very much. So hope to see you in the morning.